Okay, with a basic understanding of the electrical and chemical nature of neurons, we can start to see how they might be processing information. Let's take a look at the top diagram up here. Here we have an axon and a terminal. Here's the synaptic space here. Here's the target cell. So we're just showing the cell body. We're not showing the dendrites, just to keep it simple. And here's the target cell's axon and the target cell's terminal. Now here comes one action potential down the axon. Remember, the axon is covered with the sodium and potassium channels, and it's a domino sequence of openings of those channels. And our little icon here represents one of those little oscilloscope signals racing down the axon. Remember, the left side of the curve was sodium in, and the right side of the curve was potassium out. Well, here comes one action potential down to the terminal. Well, it's only one signal, and the effect of that is that only a few of these vesicles, right, are going to fuse with the membrane, and not much neurotransmitter will be dumped out into the synapse. Well, if there's not much neurotransmitter, not many of these channels will be opened up, so not a lot of sodium can get in. So we have a small inflow of sodium, right? Which means we may not reach threshold, so we do not open up those first key channels on the axon here, so we get no action potential. In effect, the target cell is saying the input was too weak, right? Okay, middle picture. An obvious difference, and it should make intuitive sense to you. Suppose we have multiple action potentials coming down, one right after another, and here's 3 per second, let's say, then that would be 3 hertz. So it's a frequency, right? It's a measure of frequency. How many signals per second? Well, we'll call this low frequency. But nevertheless, there are multiple signals going to be approaching the terminal. So more of the vesicles will fuse, more transmitter will be released, more channels will be opened, more sodium gets in, and we might just reach threshold so that the target cell will generate its own action potentials. In other words, enough sodium got in to open up these channels and do it twice, causing these 2 deflections here, right? So we have a response. 2 action potentials were generated in response to this pattern of input activity. Down at the bottom, you see the obvious result of high frequency stimulation of the target cell. We have lots of transmitter release, lots of channels opened up, lots of sodium in. So there's so much sodium in here, it means that many action potentials can be generated in a short amount of time. So a high frequency response. So the moral of the story here is that the activity in the target cell is dependent upon the nature of the activity of the input cell, right? You have to reach a threshold, but beyond that threshold, then the higher intensity of the input, then the higher intensity of the output signal. Now, the idea that action potentials can differ in their frequency. In other words, a target cell can make high frequency action potentials or low frequency action potentials. That turns out to be one important way that neurons can, again, convey information. The nature of the electrical signal, the frequency of the electrical signal, can carry information about the input to that cell. So for example, let's say this is a sensory cell here, and we can think of the sensory cell as transforming some stimulus in the world, maybe light, or maybe air pressure waves that we would call sound, or maybe pressure to the skin, touch. What sensory cells do is convert those external stimuli into electrical signals, action potentials. And the idea is this sensory cell, because it is responding to a soft sound, right? If it were in the ear, or if it were on the skin, a soft touch, or in the eye, a dim light, it generates a low frequency set of signals. So low frequency action potentials then, in a sense, means that the stimulus in the world is a soft one, either it's sound or touch or a dim light. On the other hand, a loud sound or a hard touch or a bright light would increase the frequency of the sensory cell's response, right? So the high frequency signal here, in a sense, means that the stimulus in the world was loud or hard or bright or whatever, depending on what sensory system you're talking about. So you see then, this is called frequency coding, that the frequency of the action potentials can carry information about the nature of the stimulus. And one final example of a neuron sort of processing information comes into play when we think about a target cell here that may have both excitatory inputs and inhibitory inputs. And so the cell's going to have to make a decision. It'll have to weigh the relative weights of the excitation and inhibition in deciding what frequency of response it's going to generate. So let's take the excitatory. So the green one up here, we're calling this an excitatory synapse. The transmitter released and the receptors here are such that when the channels open up, positive ions go in. So these would be like sodium ions coming in. When sodium comes in, that pushes the cell towards its threshold, right? So it can start to open up those channels on the axon. So it's going to excite the target cell when positive charges come in. That's why we call this an excitatory synapse. But down here we have an inhibitory synapse. So the identity of these neurotransmitters with their receptors, remember the transmitters are like a key fitting a lock, so they kind of work as a pair here. But this pair of transmitter and receptor, it allows chloride ions in. Chloride ions are negatively charged. If negatively charged ions get in here, well, that prevents the cell. That has a tendency to prevent the cell from generating its electrical signal, right? Because if the interior gets more negative, then these channels over here will not open. So there's a battle between the excitation and the inhibition here. The inhibition can cancel out the excitation, in which case the target cell will not generate a response. But if the excitation overwhelms the inhibition, then the target cell will make a response. If the inhibition overwhelms the excitation, the target cell will stay quiet. So here we see the conditions and the results here for, if the excitation is high, you get high frequency signals in the target cell. If it's low, low frequency signals, or maybe none at all if it doesn't reach threshold. If there's only inhibition, then you're going to get very low frequency signals because sometimes, there are just sort of random signals that are generated in these living cells. And then, of course, here's the integration aspect of the target cell. If you have a combination of excitation plus inhibition, then the output signal, the frequency of the output signal is going to depend on the relative balance between the excitation and inhibition.